So the nephron is the structural and functional unit of the kidneys. What the nephron does is actually take the blood plasma out of the blood, reabsorb everything that you want to keep, and what's left over is the waste products that you want to get rid of. If you're looking for your keys in your purse, you dump your whole purse out and you put everything but the keys back in. That's the most efficient way of separating things out. The kidneys do the exact same thing. What happens is the blood plasma is filtered out of the blood, and then it's run through. This filtrate is run through a series of tubes, and in the tubes we reabsorb everything that we want to keep. So this is all accomplished by the nephrons. There's a million per kidney, a couple of million of them in each of us, because we have two kidneys. And each one does the same thing. And so the total function of the kidneys is the sum total of what each nephron does. So there's two main parts to it. One is the glomerulus, and this is the group of capillaries that actually filter the plasma out of the blood. It's the place where filtration happens. And then the much larger part, which is called the renal tubule, that is all about reabsorbing that which we want to keep back into the body. When we look at it, it ends up looking like this. So the capillaries here in the glomerulus is where the filtration happens. The tubes that collect the filtrate and then reabsorb is this, and these are the nephron tubules. It consists of Bowman's capsule, or the glomerular capsule here. The glomerulus and the glomerular capsule together are called the renal corpuscle. This, which is the proximal convoluted tubule, which means the twisted tubule close. This loop that Dr. Henle named after himself, the loop of Henle. Then the distal convoluted tubule. And the distal convoluted tubule ends up attaching into a duct called the collecting duct. And as you can see from this picture, that in like this particular collecting duct, there's 1, 2, 3, 4, 5, 6, 7, 8 nephrons dumping into this particular collecting duct. So the collecting ducts collect the product of all the nephrons. The stuff that's in the collecting duct isn't urine yet, but it's close. So it starts out at the renal corpuscle, the glomerulus and the glomerular capsule. And in these capillaries in the glomerulus, they are fenestrated capillaries. They are very leaky capillaries. They allow liquid to pass from the blood plasma into the space that is in the capsule. It's kind of like a coffee maker. The coffee pot that collects what comes through the filter is the glomerular capsule. The filter basket, where that keeps the grounds and only allows the liquid to pass through, is the glomerulus. The glomerulus has all kinds of holes, the exact same way that the coffee filter has holes in it. Now, the capsule has got two layers. One layer is called the parietal layer, and it's a simple squamous epithelium. It's basically the outside of the carafe that's going to catch the filtrate. The layer that's actually adhered to the capillaries of the glomerulus is called the visceral layer. And they have specialized epithelial cells called podocytes. And podocytes are really these kind of octopus shaped cells that literally wrap around the the capillary and have and therefore create slits that allow the filtrate to pass through and into the space between the visceral and parietal layer. So this is the parietal layer. As you can see, they're just squamous epithelial cells with tight junctions that form a barrier. Wrapped around the capillary, the fenestrated capillary, see the little holes there, are the podocytes. Between the two is a basement membrane, a layer of extracellular material that kind of glues the two things together. We'll see in a minute, actually, that the basement membrane, the fenestrations, the basement membranes, and the filtration slits of the podocytes together make up basically the filtration membrane. That's what the plasma has to pass through in order to get into the capsule. So when the filtrate, because it's not blood plasma anymore, it's actually blood plasma without any of the large chunks, the grounds. So the proteins and the cells and the platelets and all of that stuff stay in the blood, and the only thing that comes through is the liquid plasma and whatever is dissolved in it. So the liquid's collected by the capsule, Bowman's capsule, and then it flows into an area called the proximal convoluted tubule. Now, the walls of the convoluted tubule, the PCT, are cuboidal cells, and they have all kinds of microvilli on them for surface area, and they have a lot of mitochondria because they are very highly metabolically active. They use a lot of ATP, and its main job is reabsorption. So what starts to happen here in the proximal convoluted tubule is we start moving things from the filtrate back into the blood. It's the things that we want to keep. So this would be things like sugars and vitamins and ions and the things that we want to keep. What we don't want to keep are things like nitrogenous waste, ammonia, urea, that kind of stuff. There is a little bit of secretion happens in here, but we'll talk about that later. So they end up looking like this. The reason that there's so many mitochondria is because a lot of what causes the reabsorption is active transport. It's the sodium-potassium pumps and such. So very, very highly metabolically active. And that's one of the reasons why the kidneys are so very good at detecting low oxygen levels, because they stop working as well when there's not enough oxygen to make ATP. The loop of Henle is the first part of the renal tubule that actually descends into the medulla. All the renal corpuscle and the proximal convoluted tubule are all in the cortex. The loop of Henle, ascending and descending limbs, pass down into the medulla. And there's two parts to it. The descending limb is thin. It's a simple squamous epithelium that's permeable to water. And a lot of water reabsorption happens here. The ascending limb has got a cuboidal epithelium that's not permeable to water, but has a lot of sodium-potassium pumps, and so is used to reabsorb sodium. So the thin segment looks like this, right? And the thick segment looks more like the proximal convoluted tubule. The distal convoluted tubule is where the final reabsorption happens. By the time the filtrate gets to here, almost all of the nutrients and things that we want to reabsorb are reabsorbed. The distal convoluted tubule is all about reabsorbing more water, especially if you're dehydrated, and sodium, and getting rid of potassium if we have too much potassium. It's probably got more to do with secreting potassium and getting rid of the excess potassium than really absorption. Now, this again is found in the cortex, not rather than the medulla, and the cell walls look the same. And you'll notice that they look a lot like the proximal convoluted tubule, lots of mitochondria because metabolically active sodium-potassium pumps. Now, the collecting duct receives the filtrate from numerous nephrons, from the distal convoluted tubules of a number of nephrons. And they actually carry this filtrate down towards the papilla in the renal pyramid. They fuse together, and more water reabsorption happens here if need be. It's, like I say, if you're dehydrated especially. And when it's finally through the collecting ducts, it is now urine. The walls of the collecting ducts are made of cuboidal cells, and they have microvilli because it's all about absorption. And they really are very useful in acid-base balances, so it's bicarbonate and that kind of thing, and dilute versus concentrated urine. It kind of looks like this. There's a couple of different types of cells, but they're all basically cuboidal. The principal cells are about water and sodium. The intercalated cells are about pH. So if we pull it all together, it ends up looking like this. So blood is delivered by this artery. This is the afferent arteriole. There's the glomerulus. There's the efferent arteriole. The filtrate is formed here through the filtration slits in the podocytes and collects in between the parietal layer and the visceral layer of the capsule. The filtrate moves into the distal convoluted tubule, or the proximal convoluted tubule rather, where we reabsorb the things we want to keep. For instance, glucose. It then moves down into here where water is reabsorbed, and then up here where sodium is reabsorbed. Then into the distal convoluted tubule, where if we need more water, we reabsorb more. If we need more sodium, we reabsorb more, where we secrete excess potassium. It's brought here into the collecting ducts, where we bring back more sodium, more water if we need, but only if we need. We adjust the pH so it's bicarbonate ions, and then it flows out here into the renal papilla, where it's picked up by the minor calyx and is now out of the body and is urine. Now, most of the nephrons are found in the cortex. The loop of Henle only just barely makes it down into the medulla, into the medullary pyramids. We call them cortical nephrons. They work the same almost all the time. The last 15% of them are found right along the edge of the medulla, so we call them juxtamedullary, which means beside the medulla. and they have very long loops of Henle's, and that go deep into the medulla. And this 15% are used to create concentrated urine when we are in a dehydrated condition. They're very important for water reabsorption. There. Now, around each of these tubules are capillary beds. Around the proximal convoluted tubule, the distal convoluted tubule, there are capillary beds called peritubular capillaries. And this is what the stuff gets reabsorbed into. That's how it re-enters the bloodstream. Now, there are capillary beds that surround the loop of Henle, and the capillaries tend to be straight. So we call this group the vasa recta. Again, the vasa recta is used more to make concentrated urine. So, the glomerulus is a capillary bed with fenestrated capillaries. So they're very leaky capillaries. They're fed by an afferent arteriole, and they're drained by an efferent arteriole. They are modified for filtration. The blood pressure in them is high because we want lots of filtration to happen. We want that high hydrostatic pressure to cause the liquid to leave, the plasma to leave. The afferent arterioles are smaller in diameter than the efferent. The peritubular capillaries, very porous, and they're adapted for absorption. And they're right on the renal tubules in the cortex. And they're fed by the efferent arterioles, by the blood that's been leaving the glomerulus. The vasa recta, as we saw, long parallel to the loop of Henle. And we find them in the juxtamedullary nephrons. And they function to make concentrated urine. I don't know what it means, information. They function in formation. There should be a space in there, in the formation of concentrated urine. Again, same picture. One of the things that happens because of all these capillary beds, blood coming through the kidneys goes from, because it's right off the aorta, it has a mean arterial pressure of about 95. By the time it actually makes it into the venules and the veins, returning to the vena cava, we're down to about 8. So there's a huge loss in blood pressure across the kidneys because of the amount of resistance. And that resistance is what causes the filtration to happen. It's interesting because early in the kidney, the resistance is modulated, or modulates the pressure. So almost no matter what your blood pressure is, the pressure within the kidneys is about the same. So if the systemic blood pressure varies, the kidneys still get about the same. And you want the resistance to also get rid of the pressure, so that in the peritubular capillaries the blood pressure is low, and more absorption will happen. In each nephron, there's an area, and it's called the juxtaglomerular apparatus. So it's right beside the glomerulus. And it happens right where the distal convoluted tubule doubles back and passes up between the afferent and the efferent arterioles going into the glomerulus. So it regulates the rate that urine is formed. There are special cells that are called JG cells, juxtaglomerular cells, and they are smooth muscle cells of the arteriole. There's mechanoreceptors that sense the blood pressure. There's an area called the macula densa that are chemoreceptors that sense how salty the urine is. And then there's extraglomerular mesangial cells that are involved in signaling. It fundamentally looks like this. So this is the afferent arteriole going into the capillaries here of the glomerulus. This is the podocyte. This is the capsular space. There's the parietal layer of Bowman's capsule. This is the proximal convoluted tubule. The filtrate goes down through here, loop of Henle, back up through all the distal convoluted tubule until it gets to here. This is the distal convoluted tubule. Now what happens is, if there's too much filtrate being formed here, then it passes too quickly through the tubules, not enough reabsorption happens, and we have more liquid here. When the more liquid is here, or that liquid has got too much salt or sugar in it, that means that this is working too quickly. So what happens is the stretch in here causes these cells to cause constriction here to reduce the pressure going into the glomerulus, so that less filtrate is formed, so that everything moves down the tubules more slowly, more is reabsorbed, and by the time that gets to here, that filtrate gets to here, there's less of it, which means there's not as much stretching, not as much pressure, which then causes this to relax and dilate, allowing more blood and more pressure going in here. So it's an automatic self-regulatory feedback mechanism that that maintains the right blood pressure in the capillaries to produce the right amount of filtrate per unit of time.